Okay, good morning, everyone. I'm here to talk about waste. Yeah, as mentioned in the title, production of bio-oils from waste-fed microorganisms. Now, the focus of this project are the bugs in these wastewaters that we are producing. These bugs are the term for them, but they are microorganisms. But most people working in the wastewater treatment field, they use bugs to call these microorganisms. Now, these wastewaters that are being treated here in this wastewater by the microorganisms are the water that we are disposing or flushing in our sinks. You know, the bathroom sink, the kitchen sink. And so these microorganisms treat these wastewaters. And now I'm going to present how we are trying to incorporate another purpose for these microorganisms instead of just being the cleaners of our waste. So here's the outline of my presentation with these different sections: the cleaning bugs, the fat bugs, the valuable waste, the oily idea, experiments, the findings, and the activated sludge lipid technology. So this project is focused on these microorganisms, the bugs, in our wastewater that are treating, in the wastewater facilities that are treating our wastewaters. And they are focused in those biological treatments in those section of our wastewater treatment plants. Now, the idea here is that these microorganisms assimilate organics from our wastewaters to reduce the VOD, COD, and other organics in their nutrients in order for the wastewater to be a treated water to be disposed in our rivers, bayous, and lakes, and other bodies of water. Now, in the process of treating our wastewaters, these microorganisms grow. And in the process of growing, there are these different classifications of microorganisms that are growing. We have bacteria, archaea, fungi, algae, protozoa, and there are even viruses, helminths, and we call them the bugs. And there are also other non-organics in that so-called activated sludge, the consortium of microorganisms. Now, before this biomass of activated sludge and other sludges from our wastewater facilities are being disposed out of the facility, they need to be stabilized. And there are two ways: the anaerobic digestion or the aerobic digestion to stabilize them, producing the biosolids. In the United States, we're producing around 8 million tons per year of these biosolids. Now, one good thing about this was that researchers back at Mississippi State, decade back, well, that included Dean Zappi, found that we can produce oil or lipids from these microorganisms. I'm going to be interchangeably using oil and lipids terms. At 5 to 10% of their weight. That's the amount of oil that we can get from them. But there was a problem. That amount of oil is not economical. We have to increase the lipid content or the oil content. And so they furthered that research, and so they went into the area of fattening the bugs, the microorganisms. And the idea, the basis for the lipid enhancement was on the pure culture microorganisms. It so happened that researchers centuries back have been working on these pure culture microorganisms to produce oils. And by utilizing the principle of high carbon to nitrogen ratio, high CN ratio, greater than 70, that's a mole ratio, the microorganisms can be induced to accumulate these lipids. I'm going to make an analogy to the human scenario. It's like the idea of taking in a lot of carbohydrates. If you take a lot of carbohydrates, you get fat. And that's also the principle for the microorganisms. If you feed them with a lot of carbon sources, they get fat, and there can be the sources of the oil. Now, but there's another problem with this stage or developing in this research, because even though this research established this idea of enhancing the lipid content, they started with sugars, and sugars are so costly, and so we had to find an alternative for the carbon source, so a non-sugar carbon source, and that's the problem that this project tried to answer. And so what I did was look at the literature and search what had been going on. And there are three main research here that have been going on, still going on, that were combined to come up with the idea for this project. The idea was that the lipid accumulation that I just discussed have been successful, and that pure culture microorganisms can utilize the so-called short-chain fatty acids. Short-chain fatty acids include acetic acid, propionic acid, and butyric acid. Acetic acid is a component of vinegar. Now, they can be produced using anaerobic digestion in a wastewater treatment facility or other facilities utilizing anaerobic digestion of organic waste. So they can use those as carbon sources instead of using the sugars. And that's a good news to me that time, because these three ideas can be combined together to come up with the principle of lipids from short-chain fatty acids, like acetic, propionic, and butyric acids, via the activated sludge process, hoping that the microorganisms in the activated sludge will perform like they're performing when they're being fed with sugars or glucose. Well, as you should expect, it was successful. So I'm going to go to the details of that. And the schematic for the whole project was this. So we envisioned that what if you make sewage microorganisms produce oil from waste? So we are utilizing these organic waste, lignocellulosic biomass, organic municipal waste biomass, and perhaps bioprocessing residuals from the same process as the source of the carbon, then produce those, or to convert those into intermediate carbons, those are the short-chain fatty acids, and feed those to the activated sludge microorganism in an aerobic process that's needed, aerobic process, to enhance the oil content, again targeting for a more economical lipid technology from activated sludge. The higher the oil content, the better. And so, basing on this idea, we set up different experiments, but this is the common procedure for all the experiments we did. Hundreds of treatments, this is a common method. Sampling of the activated sludge from a wastewater treatment plant, that was me. The bioreactor setup is a 5-liter bioreactor in the chemical engineering lab, feeding them with the different acids, acetic, propionic, and butyric acid, and after that, sampling and analysis, then further analysis. So that's the whole story that I did for 3 to 4 years. And here are now the findings. I'm going to show you first a graphical result so you can appreciate more of what happened. So this fluorescence microscopy image from the raw activated sludge that we use and the lipid enhanced activated sludge that we use. And those green regions are the neutral lipids. We targeted the neutral lipids because those were the easy ones to be converted into more useful products, like, for example, biodiesel. And as you can observe, there are more green regions in the lipid-enhanced activated sludge. That's now an initial indication that we were successful in enhancing the lipid content or oil content of these activated sludge microorganisms by feeding them with these acids. Specifically for these images, we used the acetic acid as the carbon source. So imagine like vinegar. We fed that to these microorganisms and they were able to utilize that as a carbon source. And also, we did a magnification of these images with the overlaying of the transmitted light image, just to make sure that these green regions are within the cells. And as you can see here, these bright regions, they're within the cells. And one more interesting thing about this image, the bottom, is that the size range of these cellular bodies containing these bright green spots, or the neutral lipids or oils, are in the size range of yeasts, fungi in short. Well, there's a connotation that, or an assumption that activated sludge is dominated by bacteria. And the bacteria are there, there are also fungi in there. But in here, the ones contributing to the lipid accumulation are the yeast, in the size range of yeast. So we further that finding by conducting a DNA sequencing and analysis of the samples in order for us to quantify, observe the behavior of the fungal community. And this figure is the plot of the abundance of the fungal community in terms of the phylum level, with the raw and enhanced, lipid enhanced of 3 sludges. And these 3 sludges were taken from the East Wastewater Treatment Plant along Pinhook, Ambassador Caffery Parkway along Ambassador, and the South Wastewater Treatment Plant near the Lafayette General Hospital. So they were from different places treating different wastewaters. And there was a common behavior that even though they started at different profiles at the level of the phylum, they converged into a certain phylum group, the Ascomycota. And we even went deeper into that one, and we organized the numbers in that enhanced activated enhanced activated samples. And we found this profile of those Ascomycota phylum. Now the interesting thing here is that in the order level, the Saccharomyces satiles, those are the budding yeasts. Budding yeasts proliferated during that time of lipid enhancement. So they survived and they ate those acetic acid and even other acids in the process. And this is a good finding in terms of the literature. I'm talking about the literature right now because no one haven't deliberately studied the change in the profile of the activated sludge in terms of fungi, especially the budding yeast. Now we furthered that with a target of the biodiesel as a product. And we usually measure the biodiesel profile that we are producing in terms of the so-called fatty acid methyl esters, or FAMEs, abbreviated as C16, C16-1, C18, C18-1, C18-2, and C18-3. And the point of this figure is to show you, at the bottom, is a correlation of the profile. The story here is that as the microorganisms accumulate lipids, already shown in the previous slides, the profile of the lipid is approaching a certain profile, indicated by the increase of the correlation coefficient, again tested at a population correlation of 0.9 and 0.95 at 5% alpha. And that says that the biodiesel profile that we can get from the microorganisms in their lipids is homogenizing, even though they are starting from different plants. Take note, AS2 and AS3 came from different plants. But if we try to enhance the lipid content, perhaps in a centralized facility, we can come up with a converging lipid FAMES profile. Now, this is the kinetics of all that processing. I'm going to go to show more data right now. The first point here is that, oh, by the way, this is a figure of the acetic acid profile. This is carbon source, then the pH, then the biomass, and the lipid content at the bottom. Now, the first point here is that acclimation occurs in the first time axis is at the bottom. In the first 24 to 36 hours, the microorganisms were trying to adjust. In the first 12 hours, actually, they did not consume any acetic acid. They did not. They couldn't. But after 12 hours, they started to consume the acetic acid as carbon source. Then further that, they were even able to make the acetic acid concentration almost zero. Now, that also was corresponded by the biomass growth. After that first 12 or 24 hours of acclimation, the biomass then started to increase. And of course, the lipid content increased. And that was the second point. Now, the third point here is that the carbon source conversion as compared to the previous studies. And I'm comparing here in this project the conversion or the carbon source needed per kilogram lipid. For this project, we were able to make the activated sludge microorganisms to use 23 to 31 kilograms of the acetic acid as a carbon source per kilogram lipid. And if you compare that in the previous works on sugars, they were producing or using 100 to 140 kilograms of acetic acid per kilogram lipid. So we did not just substitute the carbon source with an acid that can be produced through anaerobic digestion. We also surpassed the performance of conversion with 4 times, 5 times magnitude in the conversion of the carbon source into lipid. And this is also good news to the scientific community. Well, as indicated by the quick acceptance of the papers published with these results. Now, with this good news that I have right now, there are still some struggles with this research area. I'm showing here the costs. We did not do costing for this project because the costing part is very complicated, and I hope that some faculty right now, or perhaps Dr. Hernandez or Dean's up here, thinking about dedicating a student to do the costing. But the costing of this process is very difficult because we're dealing with sludges, anaerobic sludge to produce the short-chain fatty acids and the activated sludge to accumulate the lipids. So far, we don't have enough models, theoretical models, to simulate those. Okay, so I'm borrowing here some numbers from previous literature sources to show the numbers. For the first part, the production of the short chain fatty acids, one study conducted or focused on the production of short chain fatty acids from food waste. And that's an interesting one, the food waste, because they are high in organics. And also in this paper, they were able to show that we can go produce cheap short chain fatty acids from our food waste. That's why I picked the paper. And after producing the short chain fatty acids, they're being fed to the microbial consortia. Now, in this study too, they focused on a single microorganism, a pure culture microorganism, to have a good model establishing the costing. And they were able to show that we were just going to spend 1.05, in short $1 per kilogram of lipid if we use the short chain fatty acids from food. And that's a lot cheaper as compared to the soybean oil or castor oil, or even the microbial lipid or oil taken from sugars, if we start with the food waste derived short chain fatty acids. Now here's the challenge in this part, the biodiesel from activated sludge lipids. Because we will be spending a lot, around $3 per gallon of biodiesel. This was a study in 2007. Now, the interesting thing with this study was that this study was actually the one that sparked the idea of this lipid technology from activated sludge. But that time, the costing was not really good. And as of now, researchers are trying to find a way to go about this one. Now, another implication of this is to think about where to go next. Well, this study already proposed an innovative process called the in situ transesterification. That is an intensified process combining the extraction and production of biodiesel. But still, that was so expensive. Perhaps we have to go somewhere else. That's one of the questions right now. And so I'm going to go to this direction of this research as of now. So the activated sludge lipid technology, in the past decade, we have accomplished this: the lipid extraction and the biodiesel production from that lipid. That's started by Dhan and colleagues at Mississippi State. Then the use of sugars as carbon sources in 2010. Then right now, the use of these short chain fatty acids derived from waste for the enhancement of the lipid content. Now, where do we go from here? I'm going to give some points on where to go, my opinion, and I think backed by some facts. For example, the first point, the non-fuel applications of activated sludge or activated sludge lipids. Perhaps we can use those lipids or oils for paints. We don't eat them, we put them in the walls, so we don't have a lot of concerns about the safety and health issues. So perhaps we can use the oils in those applications. Another one is the oil and gas well drilling fluids. This is actually the topic of the next presenter, Gopi, later on. That's why I highlighted it. It's already ongoing, and I think that's his ticket to graduation this semester. So that application too. The second point is the wastewater treatment. Take note, this idea started from waste treatment. Right now that we were able to see that these microorganisms can assimilate carbon sources that were originally not their carbon sources out there in the conventional wastewater treatment facility. Perhaps right now that they're acclimated, they can utilize some resilient contaminants that as of now, the health sector is having problems. And there are a lot of them out there, accumulating in our bayous, rivers, lakes, that cannot be treated in our wastewater treatment plants. Hormones, drugs that can be treated in wastewater treatment plant. Perhaps they can right now. They are acclimated, adjusted to these conditions. The third point is animal feed applications. And I'm talking about activated sludge from the treatment of wastewater from fruit or vegetable processing, to minimize the metal content in this activated sludge. Perhaps that's also a way. And the fourth point here is that I'm borrowing this from Dhan, the base case for gaseous carbon source. So this study focused on carbon source in the liquid phase. perhaps the carbon source can also be a gas. In this one, the so-called methanotropes. Perhaps they're also in the activated sludge. The methanotropes, the idea here is that we can use methane that we are producing right now in wastewater treatment plants, feed those to the microorganisms in activated sludge, and use that as a carbon source to accumulate lipid. That bypasses the concern right now in a lot of cities trying to implement the so-called power and heat generation from biogas, from wastewater treatment plants. The economics of that is not so good. That's why not many of our wastewater treatment plants right now are just burning up, flaring up the biogas that's being produced in the biosolids treatment. If you want to install a machine right now to utilize that, it's going to be a power or heat. But the question now is, it's a brilliant idea. What if we use those biogas as carbon source for these so-called methanotropes? Perhaps they are in the activated sludge. We don't know yet, but that's a possible direction. So, with this research that I've done so far, I hope that every time that you flush water in your sink, bathroom, or kitchen right now, you think that there are some guys like me focusing on the utilization of the organic waste, swimming with those flushings you're giving. But I hope that doesn't encourage you to flush more. Let's conserve water. But yeah, we are the guys trying to utilize our wastewater treatments. Thank you.
All right, great job, Dan. Judging panel?
Okay. Terrible because I know too much about this. To me, a thing that I said this to you before. You should emphasize a little bit more that when you do the enhanced AS, that you're finding that it's a particular classification of species, you're calling it bug, that's most important that other people haven't realized. And that particular species should be applied to some other kinds of things. And that application may turn out to be as significant as some of the things you talk about in terms of other uses that you have. So the kind of questions I have to ask are more about that species, because it comes all the way through still a sugar-based species. It's a species that likes sugar-type materials. So again, is it because these products, the acetic acid, started from a sugar, a carbohydrate, to go to acetic acid that's important? Or is it the fact that a sugar potentially could be used?
Yeah, that's actually a great question that unfortunately Dr. Andre of biology can handle. I cannot answer that. It's more on the microbiology and biology aspect of it, the biochemical process. But that's actually an interesting point that I tried to put that in my outline, I forgot. The species identified as the budding yeasts, they can be used for the so-called lignocellulosic biomass hydrolysate. Those biomass, like grass or trees, that being hydrolyzed through base or acid treatment or high temperature, high pressure treatment, they usually produce acetic acid as a main product. Even though the target was to produce sugars, acetic acid was the main product. So in one of the papers that we published, the proposal was to perhaps these microorganisms that already acclimated to these as short-chain fatty acids can now be used, even isolated if you want to, but perhaps they can be used even as a consortia to now utilize these, they call them the inhibitors in the lignocellulosic biomass hydrolysate. Yeah.
And you said the 2007 study, forget what it was, with a projected cost of $3.11 per gallon. That's almost a decade old. Has anything new been published, or can we estimate what that figure would be if we could make this process more efficient?
Yeah, unfortunately, there's nothing new right now. Now, here's one thing about that. It was an innovative idea that intensified in situ transesterification, combining the extraction of lipid and production of biodiesel. It was an innovative idea. And so that year, it was one of those most cited papers, and even the following years. And I was surprised that no one followed that up with more refined, reduced cost implementation of it. So no, unfortunately, none.
One more question. This is somewhat theoretical. Okay, let's project out in the future and this process becomes commercial, you can compete with conventional oil production, which you have to drill for. And has anybody figured out, okay, if we do produce this type of oil, is the environmental impact, you know, how much carbon you release in the atmosphere, things, does it differ? Would it differ from the conventional oil?
The bottom line of this is that it's trying to emulate the idea of carbon capture. But, well, I want to be honest.
I that question very well.
Yeah, no, I want to be honest, but in the process of extracting and production of, for example, of biodiesel, we also spend some energy utilizing, for example, gasoline or coal, electricity. So those are a lot of some of the many factors that must be accounted in order to come up with a good economics. I tried economic analysis, but I do want to propose a lousy economic analysis, because again, I couldn't comprehend the a lot of factors and modeling involved. So yeah, there are a lot of factors involved, and so as of now, right now, I cannot confidently say that we are reducing or performing better in terms of the carbon capture or even emissions reduction. Perhaps we are.
We have a question, we'll allow.
The 2007 paper was using direct sugar, or what is the source for the modeling to get to the value of $3?
That paper started with activated sludge containing that range of 5 to 10% weight by weight oil in the biomass. And the idea there is to quickly produce biodiesel in situ without the extraction of the lipid, and then using that lipid to produce the biodiesel. It should be in place, production of the biodiesel. That started with activated sludge with the lipid already in place.
Yeah. And so that leads to the next one. Have you tried to sort out to see mechanistically, again, this I know is sometimes outside where the chemical engineering is, and it comes more into the chemistry. And because if you start out with something like acetic acid, you have 2 carbons in it. You know, which of the carbons is generating what particular species, and or is the species important for the actual biodiesel reaction? The reaction between the fatty acid and, quote, methanol, or is it acetic acid to get to a FAME?
Yeah, and actually that's an interesting point that I'm still trying to think about it, that if it's going to be done, it's going to be an innovative idea. And just to give you a hint about that, it can be you might be into something interesting, because this profile of the acids, they were treated with the same treatment conditions,
right,
the acetic acid feeding. But I haven't shown here, but they're published in the other papers, that if you feed them with a combination of the 3 acids, acetic, propionic, and butyric acid, there's a different profile of the FAMEs.
But what I'm getting to is with this, again, if you work with the chemistry department, you could possibly use a labeled acetic acid. So you could have like the CH3 labeled and the C that's part of the acid not labeled. And if you put that in there, you could get an idea of where more of the labeled material ended up. So if you had a high concentration of the labeled material, then you know that it was the CH3, but if it came out even, then you know both molecules are being used. And to me, that's an extremely important thing to find out, particularly when you've identified an organism that is good at this particular, reaction. Okay?
Yeah.
Let me stop it as a good comment and ending point that we agree on. And good job, Dan. Let me invite Gopi to come up and load his presentation. While we're doing that, let me mention Dan, since he got his PhD in August, he's now working at UL at the Energy Institute of Louisiana. He's on a project funded by Clico Power Company. So we were thrilled to keep Dan on